fazemos essa fala primeiro, então?
Isso. Aí você vai ficar mais tranquilo.
Aí, em vez de compor a mesa de honra, a gente vai para a apresentação do senhor.
Licença.
Para abrir, fazer a apresentação, iniciar as atividades do evento, convidando. Isso. Para iniciar as atividades do nosso evento, convidamos. Estou só esperando começar a transmissão. É que a Jordana falou que o pessoal precisa ir embora.
Tá, mas ainda não está o quê? Ainda não. Você me dá o quê?
Não. Você consegue mandar para Rafael e Biribira aí?
Mando, mas só que... Não,
Informamos que vamos começar o evento dentro de alguns instantes, só estamos aguardando iniciar a transmissão pelo YouTube. Senhoras e senhores, boa noite. É com grande satisfação que a Universidade Estadual do Oeste do Paraná, UniOeste, por meio dos cursos de graduação em farmácia, curso de pós-graduação em ciências farmacêuticas e a Universidade Tecnológica Federal do Paraná, por meio do curso de pós-graduação em tecnologia de alimentos, recebem a todos os participantes e autoridades presentes neste 10º Congresso de Ciências Farmacêuticas do Mercosul, 10º Simpósio em Ciências e Tecnologia de Alimentos do Mercosul, realizado em conjunto com o 9º EPPG-CF, Encontro de Pesquisa e Pós-Graduação em Ciências Farmacêuticas da UniOeste, e 11ª JoFarma, Jornada Acadêmica de Farmácia da UniOeste. Chegamos à 10ª edição de nosso evento. Nessa oferta, em formato híbrido, com um público de 300 inscritos, constituído por estudantes de graduação e pós-graduação, profissionais, professores e pesquisadores de diversas áreas do conhecimento, principalmente das ciências farmacêuticas e da ciência e tecnologia de alimentos do Brasil e Paraguai. Nesses 3 dias de evento, estaremos oferecendo 22 palestras, 7 minicursos e 12 comunicações técnicas, com um total de 343 trabalhos científicos avaliados. Pela terceira vez, e com muita alegria, Este evento apresenta o 10º Simpósio em Tecnologia e Ciência de Alimentos do Mercosul, organizado em conjunto com a Universidade Tecnológica do Paraná, coordenado pelo Programa de Pós-Graduação em Tecnologia de Alimentos dos campi de Medianeira e Campo Mourão do Paraná. Para dar início ao nosso evento, convidamos a todos os participantes para assistirem à apresentação cultural do Coral da Universidade Estadual do Oeste do Paraná, Campus Cascavel, e amigos do Coral do SESI, para a abertura de nosso evento, que vem trazer mais brilho a este momento. Ao Coral da UniOeste e amigos do Coral do SESI, agradecemos imensamente pela linda apresentação que acabamos de assistir, estendendo a todos os membros os nossos cumprimentos, na pessoa da maestrina Giovana Galvão Lubick. Neste momento, nominaremos as autoridades e convidaremos para a composição da mesa de honra. O diretor do Campus Cascavel, professor doutor Geisler Roges Flor Bertolini. A professora doutora Ligiane Lourdes da Silva, neste ato, representando a diretora do Centro de Ciências Médicas e Farmacêuticas, professora Mônica Tereza Suldowski. A coordenadora do 10º Congresso de Ciências Farmacêuticas do Mercosul, professora doutora Poliana Vieira da Silva Menoli. A coordenadora do 10º Simpósio em Ciência e Tecnologia de Alimentos do Mercosul, professora doutora Eliane Cola de Andrade, da Universidade Tecnológica Federal do Paraná, campus Medianeira. O coordenador do 9º EPPGCF, Encontro de Pesquisa e Pós-Graduação em Ciências Farmacêuticas da UniOeste, professor doutor Rafael Andrade Menoli. A coordenadora da 11ª Jofarma, Jornada Acadêmica de Farmácia da UniOeste, professora doutora Fernanda Coleraus da Silva. E a coordenadora geral do 10º Cosimp, 9º EPPGCF, 11ª Jofarma, professora doutora Luciana Oliveira de Farinha. Nesse momento de celebração do ensino e da ciência, gostaríamos de manifestar nossos agradecimentos a todas as instituições parceiras internacionais, amigas e apoiadoras que cooperaram permanentemente na divulgação de nossos eventos. A Universidade Nacional do Litoral Santa Fé, Argentina, a Universidade Nacional de Entre Rios, Concórdia, Argentina, a Universidade Nacional de Assunção, Paraguai, a Universidade Católica Nossa Senhora de la Asunción, campus Alto Paraná, ao Conselho Regional de Farmácia do Paraná e Conselho Federal de Farmácia por propiciarem apoio financeiro que possibilitou a participação de profissionais farmacêuticos em nossos cursos e palestras. A todos que nos apoiaram em nossa instituição na organização de nossos eventos. A Fundação de Apoio ao Ensino, Pesquisa, Extensão e Pós-Graduação, a Fundep, pelo gerenciamento financeiro do evento. Ao diretor de campus, assessores e técnicos, docentes e estudantes, que contribuíram com seu trabalho e dedicação para que cada detalhe desse evento fosse providenciado a tempo e com sucesso. A direção pedagógica do Hospital Universitário do Oeste do Paraná, na pessoa do professor Alexandro. A empresa Even 3 e sua equipe, que gerenciam e organizam os nossos eventos. Por fim, trazemos os nossos mais sinceros agradecimentos a todos os nossos patrocinadores que foram fundamentais para a realização destes eventos. Na modalidade ouro, Walters, PR Labor, produtos e equipamentos para laboratórios, Bioativa Farmácia de Manipulação e Supermercados Festival. Na modalidade prata, Azais, Herba Fitos, Inforce HT, Levamos Vida ao Seu Laboratório, Grupo MIP, Representações Tec e Sondas. Modalidade bronze, Diagnóstica Brasil. A todos aqueles amigos e parceiros, professores e pesquisadores de diversas instituições e diversas áreas que colocaram seu profissionalismo e dedicação para contribuir direta ou indiretamente para a realização deste evento, a todos o nosso muito obrigada. Sejam todos bem-vindos. Convidamos agora para fazer uso da palavra o diretor do campus Cascavel da UniOeste, Universidade Estadual do Oeste do Paraná, o professor Geisler Roges Flor Bertolini.
Boa noite. Boa noite a todos que estão aqui no auditório aqui da reitoria, aqueles que também que nos assistem, né, aqui, tanto no Brasil quanto de outros países, como já foram nominados, diversas instituições parceiras. Meus cumprimentos aqui à nossa coordenadora geral deste evento. Parabéns, professora Luciana, pelo empenho, pela dedicação. A gente sabe que não é fácil uma organização de um evento desta grandeza, né? A professora Luciana tinha o receio, quando ela foi falar comigo, sobre o espaço. Nós estamos com o nosso auditório do campus em reforma, e então ela falou assim, então vamos ter que dar um jeito. Então, acho que a solução foi a transmissão, porque é um evento realmente grande, a gente acompanha, né, 10 edições, né, Luciana? E que realmente é muito, muito trabalhoso. A quantidade de trabalhos submetidos, cada avaliação, cada palestra. Então, a gente reconhece o trabalho e a gente parabeniza a dedicação de todos vocês. Então, professora Luciana, leve os cumprimentos do campus a todos os professores, a todos os alunos e acadêmicos do mestrado, da graduação, que se envolveram e que tornaram agora a realidade esse 10º Cosimp, né? Também quero cumprimentar todos já nominados aqui na mesa, professora Eliane, lá da Federal do Paraná, muito obrigado pela parceria, pela presença aqui. E desejamos que vocês tenham um ótimo evento. Nós sempre colocamos o campus, a direção do campus, à disposição para colaborar nos eventos. Algumas vezes, algumas coisas não dão certas, outras dão, né, Luciana? A semana passada, a Luciana estava muito preocupada com a questão do coffee break, e, graças a Deus, conseguimos fechar com a empresa e deu certo, né? Então, nós sabemos que a dedicação tem que ser grande. A gente reconhece que o curso de farmácia, os professores se dedicam muito para o seu curso, para as atividades da graduação, da pós-graduação, da pesquisa, da extensão, e acho que o Cosimp é fruto disso, né? É um resultado de toda essa dedicação. Então, em nome da direção do campus, em nome da UniOeste, a gente parabeniza a todos e desejamos que vocês tenham um ótimo evento, aproveitem as palestras, as atividades, que isso faz a diferença na formação dos nossos acadêmicos. Muito obrigado.
Ouviremos agora a coordenadora do 10º Simpósio em Ciência e Tecnologia de Alimentos do Mercosul e docente da Universidade Tecnológica Federal do Paraná, campus Medianeira, professora doutora Eliane Cola de Andrade.
Boa noite a todos, aos que nos veem aqui presencialmente, aos que estão no modo online. É uma alegria a gente poder fazer parte hoje da abertura deste evento, então eu gostaria de cumprimentar a todos que estão aqui à mesa e estendo os cumprimentos a todos os participantes. Bem, nós temos a honra de representar a UTFPR nesta edição do Cosimp. Desde a 8ª edição, temos participado como organizadores, antes éramos somente congressistas. E agora temos a honra de participar ativamente e contribuir para que esse evento ele siga crescendo e se fortalecendo. Então, nós desejamos que sejam 2 dias de muito aprendizado a todos os estudantes, todos os profissionais que se inscreveram no evento e que possam usufruir de todo o material que foi preparado carinhosamente pelos nossos palestrantes. Queria agradecer também a todos os organizadores, aos colegas da UniOeste, que trabalharam muito para organizar o evento, e estendo também os cumprimentos à professora Deise, que está aqui comigo hoje, colega da UTFPR, e nós trabalhamos juntas na organização. Então, desejamos um bom evento, um excelente evento a todos.
Passamos a palavra à coordenadora-geral do 10º Cosimp, 9º EPPGCF e 11º Jofarma, professora doutora Luciana Oliveira de Farinha.
Boa noite a todos. Saúdo aqueles que estão aqui presentes no auditório, aqueles que nos assistem online pela plataforma da Event3. É com muita alegria que estamos aqui hoje, nessa mesa de honra, saudando a todos que se inscreveram e confiaram, né, pela 10ª edição do nosso evento. No ano, né, em que, no ano passado, nós completamos 25 anos e estamos oficialmente comemorando os nossos 25 anos nessa edição, com os nossos alunos, ex-alunos, professores, colegas, pesquisadores dessa instituição e da UTFPR, que muito nos alegra a parceria na realização do nosso Simpósio em Ciência e Tecnologia de Alimentos do Mercosul. Saúdo também aos amigos paraguaios que vieram, estudantes e professores que participam desse evento e que, pela 10ª, na verdade, desde a 3ª edição, né, estão conosco, prestigiando e fazendo com que esse evento tenha o status de um evento internacional. Para nós, do curso de farmácia e dos programas de pós-graduação em ciências farmacêuticas, ciência e tecnologia de alimentos, e também, né, como possibilidade de participação dos cursos de residência, que também participam enviando os seus trabalhos, né? É uma alegria muito grande termos a possibilidade de um evento em que possamos compartilhar tantos conhecimentos. Nós que somos docentes, que nos empenhamos, aqueles que estão na organização, que coordenam o evento, que estão aqui ao meu lado, os professores também que estão na plateia, que participaram das comissões científicas e de outras comissões de organização do evento, são para nós motivo de muito orgulho. Não é qualquer instituição que consegue realizar um evento dessa monta e que reúne tantas pessoas, tantos ideais, tanto conhecimento para compartilhar, né, como forma de ampliar os horizontes dos nossos estudantes, com a possibilidade de ver aquilo que é produzido de ciência e conhecimento no Brasil e também no exterior. Não é fácil, como o professor Geisler disse, organizar um evento. Sempre temos percalços, né? Mas são muito ínfimos em comparação das conquistas. E esse ano, especialmente, vi muita dedicação de todos os professores que fazem parte da comissão e também dos professores do curso de farmácia que se inscreveram para participar e dos alunos que, gentilmente, né, se apresentam aqui como interessados em receber esse conhecimento. Esperamos que vocês todos desfrutem tanto da programação presencial, que já iniciou hoje, que foi muito enriquecedora, vermos os nossos alunos da pós-graduação que estão cursando atualmente o mestrado em ciências farmacêuticas e aqueles que já foram nossos alunos e iniciaram aqui a trilha profissional de formação e aprendizado e hoje brilham, não só no Brasil, como professores de universidades, pesquisadores de outras instituições, como também no exterior. Então, para nós, professores, que estamos aqui desde o começo do curso e outros que chegaram depois de nós no caminho, que também foram nossos alunos, como é o caso da Fernanda, né? A Fernanda foi nossa aluna na graduação e hoje é nossa professora colaboradora, né? nos enche de muito, o professor Mahler, né, que também está sentado aí, nos enche de orgulho, e todos os outros professores que compõem o colegiado de farmácia que construíram a história desse curso ao longo dos anos, né? Inclusive a professora Deise, que foi nossa docente, hoje é docente na UTF-PR do campus de Medianeira, junto com a professora Elaine. Para nós, não há... vou dizer que não há dor de cabeça, né, nenhuma, né, para nós. É difícil, né, porque a gente sabe que aquilo que a gente vai colher é muito maior do que qualquer situação que a gente venha a confrontar. Hoje a Alana falou aqui, né, na palestra dela, que a gente nunca deve desistir dos nossos sonhos, né? Então, assim, o Cosimp, que começou como o primeiro congresso de ciências farmacêuticas de Cascavel, né? Hoje ele é o 10º congresso de ciências farmacêuticas e 10º simpósio em ciência e tecnologia de alimentos do Mercosul. Então, isso não foi alcançado sozinho, são muitas mãos construindo esse evento, que construíram no passado, que constroem no presente e que é um legado que deixamos para continuar sendo construído no futuro. As nossas edições seriam em anos pares, né? Porém, por questões operacionais, né, junto também do Conselho de Farmácia e outras ofertas de congresso, não foi possível realizarmos no ano de 2024. Né? Então, esse ano, realizamos no ano de 2025 e passamos a ser, novamente, um evento em anos ímpares. Né? E isso, para nós, independente da frequência, né, é um evento que já faz parte do calendário de eventos importantes técnicos científicos, se não o único em ciências farmacêuticas e o único também em ciência e tecnologia de alimentos na região, né? Que junta aqui o Brasil, o Paraguai e a Argentina. Então, hoje temos os nossos amigos participando online, por dificuldade de financiamento, né? Esperamos que melhores tempos virão nas próximas edições e que a gente possa voltar a realizar com um público enorme que enche o nosso auditório principal, né, de quase 400 lugares. Agora vai para quantos lugares, Geisler? 500? Vai continuar 400, né? Mas vai ficar mais bonito, mais aconchegante, né? Então, que nós possamos lotar o nosso próximo auditório com a nossa grande reforma pela qual a Unioeste está passando e que brevemente a gente desfrutará. Então, muito obrigada a todos, agradeço a cada um dos professores, né, que se dedicaram, a professora Poliana, o professor Rafael, a professora Fernanda, a professora Eliane e todos os professores do curso de farmácia, Prof. Verde, Prof. Mahler, né, todos os que estão na comissão científica, Prof. Deise, que nos ajudaram a chegar até aqui. Os trabalhos vão seguir amanhã. Amanhã terá início a nossa Jofarma, que ela já está passando em número ao Cosimp, né? A Jofarma já é a 11ª, enquanto o Cosimp é o 10º, né? Ela não falhou, o Cosimp falhou aí 2 edições, né, com anos pares, mas seguimos pelo caminho, né? E em breve nos encontraremos, né, para o 11º Cosimp, a 12ª Jofarma e o 10º Encontro de Pesquisa e Pós, né? Muito obrigada a todos, agradeço a cada um e que o evento seja um sucesso. Muito obrigada. Grazélia, eu gostaria de quebrar um pouquinho o protocolo e fazer uma homenagem especial ao professor Geisler. Professor Geisler, essa semana, né, a gente conversou muito, e ele nos ajudou muito, com muita boa vontade. O Geisler sempre foi nosso parceiro. Então, em nome da comissão organizadora do Congresso, eu estendo os meus parabéns e gostaria de pedir à nossa aluna do mestrado, a Luísa Voigt, para entregar uma lembrança ao professor Geisler em reconhecimento pelo esforço para ajudar a nossa edição do nosso evento aqui agora. Obrigada, professor.
Eu agradeço, acho que não merecia pelo tanto que eu fiz a professora Luciana ficar brava semana passada e nervosa, mas por questões operacionais a gente tentou se dedicar o máximo possível para tentar resolver, né, o nosso coffee break. Então, está garantido o coffee break, né, professora? Então, muito obrigado.
Agradecemos a todas as autoridades que participaram dessa solenidade de abertura, no momento em que nossa mesa de honra se desfaz. Neste momento, convidamos o mestrando do Programa de Pós-Graduação em Ciências Farmacêuticas, Lucas Carg, para apresentar o nosso palestrante da conferência de abertura desta noite.
Professor Douglas Rutledge é nascido na cidade de Brisbane, Austrália, com dupla cidadania francesa e australiana. Bacharel em Ciências pela Universidade de Sydney, Austrália. Diploma de Educação no Sydney Teachers College, Austrália. Bacharel em Química pela Universidade de Paris 6. Mestre em Espectroquímica e Análise Estrutural pela Universidade de Paris 6. PhD pela Universidade de Paris 6 com o tema Bioconversion of Terpenoidic Molecules by Bacteria of the Digestive Tract of Xylophagous Insects. Foi diretor do Laboratório de Química Analítica do Agro Paris Tech, onde também foi professor de Química Analítica. E atualmente é professor emérito do Analytical Chemistry at Paris Tech, professor adjunto da Charles Sturt University, Austrália, Université Paris-Saclay, Faculdade de Farmácia e do Museu Nacional de História Natural da França. Membro das Sociedades do Conhecimento de Química Analítica e do Comitê Científico como membro efetivo do Grupo Francês de Quimiometria. Membro dos comitês científicos dos periódicos In Vino Analytica Scientia, Chimiometrie, Chemometrics in Analytical Chemistry, editor do periódico Trends in Analytical Chemistry, consultor científico da Sociedade de Experts Químicos da França, correspondente associado da Academia de Agricultura da França, com H-Index 47, professor Douglas já publicou mais de 259 artigos científicos, 22 livros, 309 conferências e comunicações técnicas em congressos científicos, tendo recebido 34 convites como palestrante. Atualmente também colabora como professor visitante do Laboratório de Química da Universidade Tecnológica Federal do Paraná, em Campo Mourão. Convidamos o Dr. Douglas Neil Rutledge para ministrar a palestra intitulada Using the Power of Chemometrics. Dr. Rutledge, welcome.
Boa noite a todos. Antes de começar, eu quero dizer duas coisas. Número 2, eu falo português muito mal. Então, eu vou fazer minha apresentação em inglês. Desculpe. So the objective of what I want to present to you tonight is to try and convince you all, if you're not doing it yet, to start playing with chemometrics. Chemometrics is a large number of different technologies and techniques that can be used to optimize the acquisition, analysis, and interpretation of chemical data. Initially, it was just chemical data, but these methods work so well that other people are applying these methods to non-chemical data, biological data, etc. Here you can see the evolution of the use of the word chemometrics in the literature over the last 50 years. And so you can see that it really starts to become important around about 1980. Obrigado. 1980, why 1980? Because that's when you started having personal computers, because these techniques require computing power. So, chemometrics is a lot of different techniques. You can see them listed here. I'm going to talk about just one aspect, multivariate data analysis, because this is the one that has the most importance in agriculture, in food, and in pharmacy. Although I have to admit experimental design is very important as well, but I won't talk about that. So multivariate data analysis always starts off with something that looks like an Excel spreadsheet. In the columns, you've got your different variables, the things you've measured on the samples, and in the rows, you've got the samples. So it's just like a spreadsheet, an Excel spreadsheet. But the idea is that when you take this matrix, this set of data, you can imagine that you have, if you've got 3 variables, you've got a space defined by these 3 variables, a multidimensional space. And each line, each individual, is positioned in the space based on the numbers. Like here, you've got 2, minus 0.5, and 1. And so this individual is at this position, 2, minus 0.5, plus 1. Now that means that if you have lots of individuals with similar values, they're all going to be close together. If you have a trend in the values, they're going to be spread out in a particular way. This idea was introduced more than 100 years ago. This is older than Einstein's special relativity. So it was Pearson who came up with the idea, and it's a brilliant idea because, well, let's look at it this way. If in your data matrix, it's just noise, that means that all your individuals are just going to form a little spherical cloud in space, and all the different, there'll be no interesting directions. All the directions will be equivalent. It's just noise. It's spherical. So, just noise, a spherical distribution. If you don't have a spherical distribution, that means you don't have just noise. And that means it's interesting to find the directions of dispersion of the individuals. And these directions are going to tell you things about the samples and tell you things about the variables. So the basis of this technique, principal components analysis, is to find these principal components, these new variables. These are the original variables. And so we're going to calculate new variables based on the old ones, describing the dispersion of the samples. Seems like a nice, simple idea. What's good with this simple idea is that it produces really interesting results. When you do it, first of all, it's unsupervised. You don't tell the program anything about the samples. You just give the values of the variables. And by doing this calculation, you can find new variables, the PCs, the principal components, that correspond to the maximum dispersion. And these directions are interesting. And so you end up with these new coordinates here, based on the principal components. And you've also, they're called the scores. And you've also got the connection between the original variables and the new ones. These are called the loadings, and they tell you things about the variables. The scores tell you something about the samples. The loadings tell you something about the variables. I'll give you a simple example here. This is some data produced by Richard Sprague, who used to work in a company manufacturing scientific instruments. For example, infrared spectrometers. So this Richard Sprague had a simple piece of polystyrene, a piece of plastic, and he stuck it in infrared spectrometers coming out of the factory. So he had 100 instruments coming out of the factory, 100 machines coming out of the factory. He put the polystyrene in and he acquired the spectrum. So we acquired the spectrum between 4,000 and 400 centimeters minus 1. And this is what the spectra look like. Okay, this is 100 spectra. I got the data and I applied principal components analysis to it, and I'll show you the results. At the top here, I've just plotted the average spectrum. And then I applied PCA, principal components analysis, and here, these are the loadings for PC1. It shows the connection between the original variables and the first principal component. And you can see it's the same thing, which means that nearly all the variability in the spectra corresponded to the polystyrene, which is good, which means that his machines, his 100 machines, seem to be working well. Principal component number 2 is interesting. If you know anything about infrared spectroscopy, you'll immediately realize that this big peak here and this small one here, they're due to CO2. These ones here and here are due to water vapor. So PC2 is just people breathing on the machines, or changes in the CO2 and H2O in the atmosphere around the machines. So this is not a problem for Mr. Sprague. This is just variations in the atmosphere. This is interesting, this one. You've got two phenomena here. You've got these oscillations. These are called beats. They're due to interference. This sort of thing you observe when you have a change in the optical path length of the sample, which means the polystyrene was either slightly inclined like that, or slightly up or down or to the side, which changed the thickness of the polystyrene. And that results in these beats, these interference beats. You've also got this other thing here and here and here. These look like the first derivative of the peaks. Every time you've got a big peak here, you've got a thing like a first derivative. You observe that in principal components analysis when the peaks move between the samples. So that's PC5. PC6, you've got the same thing. You've got the beats, and you've got the first derivative. You can see it really well here. First derivative of this peak here. So there are peaks that are moving, and apparently all the peaks are moving. So this is a problem. This means that there's something happening with the instruments. So to make sure it's the first derivative, I calculated the first derivative. Here's the mean. I calculated the first derivative, and this is PC6. So there's a problem. There's a problem with the machines. So I plotted the scores of the samples. PC5 and PC6, these were the ones with the interferogram, with the first derivative. And what do we see? All the samples, except for one, are a little cloud centered on 0. And you've got one instrument, which is strange. This spectrometer number 61, here I've plotted 60, 61, and 62. You can see there seems to be a shift. If I amplify, here you've got 60 and 62, and there you've got 61. So spectrometer 61 is shifted. Look again at the spectra. You may have noticed, looking at the spectra, the water vapor and the CO2, but you would not have seen any of the other phenomena. The change in the optical path length, the sample that was shifted, you wouldn't have seen those. This shows that principal components analysis can bring out of your data things that you do not see with your eyes. And that's the power of principal components analysis and other chemometric methods. Now, I hope I've convinced you that PCA is a good thing. I'm now going to try and convince you that ICA is better. PCA is a very good technique. I suggest you all apply PCA whenever you can. But once you've done PCA, try and apply independent components analysis because it's even more interesting. When you do PCA, as I said, you imagine that your samples are points in a multidimensional space. When you do independent components analysis, your vision is different. You imagine that your matrix is a set of observed signals. You're looking at the lines. And each line is an observed signal which is made up of pure source signals that you don't know. And what you want to do is extract the source signals from the observed signals and calculate the percentage, the proportion in each sample. So here you've got X1, which is a mixture of signal 1 and signal 2 with these proportions. Signal number 2, same two source signals in different proportions. So what you want to do is calculate the source signals and the proportions of each one, the A and the S. Now, you do this by making a hypothesis. You take the hypothesis that there is no connection between any of the source signals. The source signals are independent. That's the origin of the name, independent components analysis. The other thing is, if you have a source signal, its histogram, the distribution of intensities, is not going to be Gaussian. Gaussian corresponds to noise or a mixture. If you mix together two source signals, the result observed signal is more Gaussian. That's because of the central limit theorem. And I'll show you here. I've calculated 6 signals, rectangular, rectangular, triangular, triangular, sinusoid and sinusoid, with 2 different frequencies. These source signals, there are 800 points in each one. I calculated 100 mixtures of these source signals in random proportions and mixed them all together with a little bit of Gaussian noise. And this is the result. Here you've got 100 signals mixed together. Now, here are the histograms. So you can see these are not at all Gaussian. You've just got 2 levels here, top and bottom, top and bottom. Here, all the levels are about equivalent or equiprobable. Same thing here. And then here, the top and the bottom are more probable in a sinusoid. So these are the histograms of these source signals. They are not Gaussian. If you mix them together, the result is more Gaussian, central limit theorem. So what we're going to try and do is take these mixtures that are Gaussian and try and extract from them signals that are as little Gaussian as possible. Just to see what the things look like, here I've plotted the 9 first rows in the matrix, the first 9 rows out of the 100. And you can see the mixtures there, rectangular and triangular, etc. And here you've got the histograms, were vaguely Gaussian. If I do a PCA on that, I convinced you PCA is a good thing. This is the result with PCA. The first 6, this is good because there were 6 pure signals initially. The first 6 are a little more structured than the last 3. This corresponds to the Gaussian noise I added. So these ones here, this is a good beginning, but it's not good because you do not have the real signals there. And they are Gaussian mixtures. Because they're mixtures, they're Gaussian. If I do ICA, I get that. So I hope you realize PCA is good, ICA is better. Triangular, sinusoid, rectangular, sinusoid, triangular, rectangular. And here we've got the histograms that correspond. So this technique is really a powerful technique which gives you results that are easier to interpret than with PCA. And I'll apply ICA to the same data and you'll see the difference. There is IC2, just water vapor. IC3, just CO2. IC4, just the change in the optical path length. IC5, spectrometer 61. So this technique helps you have interpretable loadings, signals, and also gives you more direct information about the samples. Now, that's just a teaching example. Now let's have a look at some of the real applications. This is some work I did with a company in France, which is no longer just in France. We were analyzing more than 400 different types of milk from European farms in lots of different countries. This is a very simple NMR spectrum of milk. So we're using NMR to analyze these 400 or something or other milks to try and see if it was possible to identify the different types of milk. So this work was done in collaboration with a company called Eurofins. They've been around for a little more than 25 years. I knew them when there were 2 people working there. Now they are 65,000. They've got an office in Sao Paulo. They do lots of different scientific work and analytical work. They love collaborating with universities. I've had 5 PhD students financed by them. They love working with scientists in universities. So if you ever get in contact with these people, try and set up projects of research, financing students, PhDs and postdoctorates, etc. They're very smart people. They make you work very hard, but they're very smart people and they like science. So I was working with them on these different milks. I worked on honey and a whole lot of other things as well. But here with the milk, we got the NMR spectra and we broke it up into blocks. We did a lot of pre-treatment to tidy things up. And then we applied ICA and we found lots of interesting things. This is one section in the spectrum. This is the signal that we extracted by ICA. And here I've plotted the proportions, the scores that correspond. And we've got no fat milk, 1% fat, half cream, and full cream. I just applied ICA, and without telling the program that there were these different types of milk, it found a signal that allowed me to separate out based on the fat content. This other section, we extracted a signal. It's a little harder to see, but the dark blue is normal milk. We've got green here, you can't see it, but I can, which is half delactosed. And at the top here, totally fully delactosed milk. Why anybody would half delactose milk, I don't know. That's why there is very few of them. I don't think that's very interesting for anybody. If you're intolerant to lactose, you don't just drink half of it. This one here, from another region, the signal we extracted is here, and these are the different treatments of the milk. You've got sterilized down at the bottom here. You've then got orange hidden in there, around here. These are ultra-high thermal treatment. The blue one up here is microfiltered and pasteurized. Here you've got pasteurized, and there's a dark green one here, which is microfiltered. And so just without telling the program anything about the milks, we got a signal which allowed us to characterize the thermal treatment of the milks. Then this one here, because it wasn't just cow's milk, we had some goat's milk as well. And there were these 2 signals, 2 different ICs that separated the goat's milk from the cow's milk. And this signal, we did not expect this one at all. This is a very strange signal because it's just one peak, which is rare in NMR. And we couldn't find anything among all the characteristics of the milk that we could imagine. We couldn't find anything that explained the separation of the samples. And so we identified this peak. This peak is formic acid. Normally, you don't put formic acid in milk that people drink. You can put it in the trough in a field where the calves are going to drink the milk because the mother's occupied doing something else, the cow's occupied elsewhere, to stabilize the milk so it can stay there without fermenting straight away. But apparently there are some situations where you're allowed to sell milk for human consumption with formic acid in it. And so we detected this in just a very small number of samples, about 10 samples, 20 samples out of the 400. That's an agricultural application. Here I worked with a pharmaceutical company. They wanted to use Raman imaging, microscopic imaging, to characterize pills that were being manufactured. So what they had is a Raman hyperspectral microscope. They put the pill there, and then they acquired a Raman spectrum on each pixel in the image. So here you've got the different pixels of the image, and here you've got the spectra going in that direction there. So you get this cube for one sample, and you unfold it, so you have all the pixels going down like that, and all the spectra going like this, and you apply ICA. You extract your ICs, your components, your signals, and for, let's say, IC1, You get all the pixels and you fold it back, and that gives you a chemical, between quotation marks, a chemical image. This corresponds to this. So where it's red there, that means this signal is very intense. So this is the set of 3,600 spectra from the pixels, and there are 801 wavelengths there, and we applied ICA to this matrix. The first component we got, IC1, this is the signal, and this is the folded up pixels. And so you could see that there are these different regions here which correspond to this compound. This is IC2, the signal, and this particular point here and another one there. IC3, etc. It's nice to have these images, but what's nicer is you take these signals and you consult a database, a library, and you identify the compounds. So here, signal number 1 was the active principle, and you could identify it with a correlation of 0.98. What I want you to see is the signal extracted is the blue one, and the one in the database is the red one. And I think the signal that is extracted is better than the one in the database. You can see for the baseline, for example, the baseline is much better. Why? Because this IC is extracted from 3,600 spectra. So you've got an averaging of the spectra, which gives you something better than if you just did one spectrum on a pure sample. Same thing here, we've got signal number 2, which is an excipient, and I think that the extracted signal is better than the one in the database. So this one's identified with 0.91. This one here, another excipient. You see the baseline here, which isn't straight in the pure spectrum, where it's straight in the IC. And this one's identified with 0.98. Now, all that's nice. These are spectra acquired by the company on their own pills. What they wanted to see, in fact, was whether the pills were being nicely manufactured, if you've got something nice and fairly uniform. It's important to have pills that have a uniform composition. But this is a problem that they had. They discovered that there was somebody selling their pills on internet, and they don't know how come somebody could be doing that. So they ordered some of the pills, and they applied ICA. First of all, they used a technique to determine how many signals were in the matrix. For their reference compound, their own product, there were 3 compounds in it. In the one they bought on internet, there were only 2 compounds. This is a bad sign already. The packaging was perfect. The pill looked exactly the right sort. It had the name on it and everything. And so they looked at the results of the ICA. This is the authentic pill. You've got the reference spectrum in red and the extracted signal in blue. They've identified it with 0.99, this one here with 0.99, this one here with 0.98, and these corresponded to the compounds that should have been in the pills. As well, you can see nice uniform distribution of the compounds within the pill, which means it's being manufactured correctly. The one that was doubtful, first thing, which is scary, is they're not very good quality pills. They're not very homogeneous. This is bad manufacturing. And then these are the signals that These are the signals that they extracted. They don't look much like these ones here, and so they stuck them into their database to identify them. This one here is metformin, which is a pharmaceutical product which should not be in the pills that they were selling. It was for another sickness. And this is an excipient, a fairly commonly used excipient, but not the one that's used in the real pills. And so they were able to prove that these were not authentic pills, that they were dangerous, and then they could try and get somebody to go and catch these people. I'd like to stress, do not buy pills on the internet. Huh? So that was one example in pharmacy. Another example, this is in what's called metabonomics. Have you heard of metabonomics or metabolomics? This is where you analyze using NMR or liquid chromatography, mass spectrometry. You analyze fluids, for example, tears, blood, urine, sweat, to get the metabolites that are in these liquids, and then use the characteristics of these metabolites to characterize the state, the biology, the metabolism of the people, the children, the animals, the people in general. This was a case where they wanted to see if adding an antioxidant to the food of some rats compensated for their bad eating habits. So they had rats, 3 groups of rats, one group with a normal fat level in their diet, 5%, and 2 with higher levels, 15 and 25% fat in their meal. They then added catechin to half of them and not to the others. So they're going to see if the catechin, an antioxidant, modifies the metabolism and the changes in the metabolism due to the extra fat. And so they collected the urine after 17 and 38 days and then analyzed it by liquid chromatography and QTOF mass spectrometry. I got the data. I did a log transformation on it and then applied ICA. This is an example. Here you've got the retention time, and here you've got the M on Z of the mass spectra. So I'm applying independent components analysis. I'm going to extract the source signals, and the source signals, they're like loadings, as we saw in PCA, and the proportions, that's the quantity of each source signal in each observed signal, they're like the scores. So here I'm plotting the scores. So it's exactly the same values in the 3 figures, it's just that I've colored them using the 3 criteria, fat, catechin, or time. And so what you can see is independent component 2 perfectly separates the 2 catechin levels for the rats. This one's interesting. This is time. Almost perfect separation, but there's 1, 2, 3, 4 rats who are on the wrong side. Now, if it had been an even number, it would have meant that somebody mixed up the tubes. But it's an odd number, so it's not just tubes getting mixed up. That means that there are some rats who have a faster metabolism than the average, and so they've got back to a normal state more quickly. So instead of being at the level of 17, they're at the level of 38 days. And then IC5, it's this one here. At the bottom, you've got the normal fat level. At the top, 25%. And in the middle between the two, with some overlap, you've got the 15%. So without telling the program anything about the characteristics of the samples, by using ICA, we've been able to extract signals that characterize these different parameters in the experiment. And here we can have the signals. This is the signal characterizing catechin, the one for time, and the one for the amount of fat in the diet. Another example in microbiology. This is some work I did. It was again with urofins, in part. We wanted to see if it was possible, using Raman spectroscopy, to identify the genera, the species, and the serotypes of different bacteria. And so we grew bacteria on Petri dishes, acquired the spectra, and there were 13 different types of organisms, microorganisms. We did some data pre-treatment to get rid of interference in the spectra, and then applied ICA. So here you have all the spectra. How many were there? Almost 600 spectra. And so these are the spectra plotted normally, and here I've plotted them differently. Here I've got the color corresponds to the intensity. So here, that corresponds to this point here. And this one here corresponds to this point here. And so here I've put on the side colors that correspond to the 13 types of microorganisms. And so just looking at it, you can see that there is some characteristic signals for the different types of microorganisms, just looking at the spectra. So we applied ICA. We determined, first of all, that there were 8 different signals in the matrix. And so we applied ICA with 8 independent components. And here are the results. At the top, we've got the signals we've extracted. Oh, the thing I didn't show you, you can't see it here, but that region there and that region there correspond to DNA and RNA. And so here I've colored in red the regions corresponding to DNA and RNA. They don't seem to have much effect on, they don't contribute much to the ICs. So you've got this IC here, and you can see it separates a little bit the different colonies. IC2 separates differently. You can see this one's up the top, where it's down there. This one's up the top, and it was down there. Okay, so you can see that the different ICs separate the groups differently. What's also good is here you've got, let's say, 50 different spectra on different parts of the Petri dish, and you can see that they're all fairly well lined up. You have fairly flat repetitions of the spectra for a particular microorganism. Here we've got another one that characterizes particularly this group of microorganisms, and here as well. So you can see that the different ICs are extracting different information about the microorganisms. And these 2 pieces here are very characteristic of this type of microorganism there. Now, what is important to see is that ICA does it better than PCA. Here we've got the results for particular signals for ICA, where you've got this flat separation of the different groups. Here you can see a lot of noise. Within each group, it's very, very noisy. And so it is not as effective in extracting characteristic signals for the microorganisms. As well, here you have a flat baseline and then a positive peak. Here, mainly a flat baseline and a positive peak, whereas here you've got positive peaks, negative peaks, etc. This is not normal. This is because PC loadings are mixtures. They're not pure, whereas ICA signals tend to be real signals. And if we plot it in 2D, so here you've got IC1 here and IC2 here, you can see little packets of the different microorganisms in nice little packets. Whereas when you do it with PCA, you've got all this dispersion, they're all spread out. So PCA is good, but ICA is better. So, in the comparison PCA-ICA, ICA extracts signals that are easier to interpret because they they're supposed to correspond to real signals. And it gives you a much better non-supervised clustering of your samples. This I observe very, very often. And good. I like chemometrics. Chemometrics is really good fun. And it's really good fun for many reasons. You get to play with good data coming from nice people. Another thing is that there are so many different methods being invented all the time. ICA, for example, is only about 30 years old. It's a very recent technique compared to PCA, which is 120 years old. In the same way, there's a whole new range of techniques that are being developed and becoming very popular. These are methods which don't just analyze one matrix, it analyzes a whole set of different matrices about the same samples. So you can have a matrix of NMR, a matrix of mid-infrared, a matrix of sensory analysis, a matrix of wet chemistry. You can have all these different matrices about the same samples, and you analyze them all together separately. And I've been using this for a while now, in particular one method, which was initially called Common Components and Specific Weights Analysis, which is a terrible name for a method. Usually methods are much shorter names. A colleague wrote a program, a function to do Common Components and Specific Weights Analysis. He wrote it in MATLAB, and he called it ComDim, which is much better. And so I used his program to start off with, and I've done a lot of publications calling it ComDim. So now, These people who invented this method now call it Cosimp. This method allows you to simultaneously study several matrices with different variables on the same samples. When you do this, you get a whole lot of different data that help you to understand what's happening in your matrices. To show you quickly how this works, it's very simple, very intelligent. Let's say you've just got two matrices, x1 and x2. You calculate the product x transposed, and that gives you a square matrix. If you've got 50 individuals and 100 variables, 50 individuals and 1,000 variables, this matrix here, W1, is 50 by 50. This matrix here is 50 by 50 as well. So they're the same size because you've done this product. You take the two of them and you multiply all the values here by lambda 1. This is called the salience. Initially, you set lambda 1 equal to 1. Same thing here, you multiply by lambda 2, initially set to 1, and you sum the two of them. So here you've got a matrix which contains all the information about the variability of the variables. You do a PCA, and you get the first vector of the scores of the individuals. Now this first vector gives the distribution of the individuals, a first estimation of the distribution of the individuals in the space of the two tables. But this first estimation isn't good because you used the idea that both tables had the same importance. But maybe one is interesting, the other one's not. So what you do is you recalculate the lambda 1 and the lambda 2, and you recalculate W, and you reapply PCA, and you do this until the lambdas don't change anymore. If a table isn't important, its lambda will be low. This lambda called the salience will be low, close to 0. If it's very important, it'll be high, close to 1. And so once it's stabilized, that means you've got the scores common to all the tables. And you've got a measure of the importance of each table. Once you've got that, you've got the first common component. You subtract the first common component from these two matrices, and you start all over again to get the second component, third component. In the end, you end up with the importance of the different tables. You get the global scores of the individuals. You get the loadings for each table, so you can understand what the tables are doing, what the variables in the tables are doing, and you can get the sum of the saliences for each table or the sum of the saliences for each dimension. We did a paper a few years ago, 10 years ago now. I talked about metabonomics. Metabonomics, you use NMR or LC mass to try and characterize the metabolites in biofluids. What's important is to know if you can trust the results you get, because each lab will use different methods, different instruments, different methods of treating the data before analyzing, etc. Will they all end up getting the same result? So we did a study. There were a whole lot of laboratories involved in it, mainly in France, but some in England as well. So what did we do? 15 members of the laboratory and their relatives supplied urine samples. The urine samples were just kept as is, or metabolites were added. How many? 32 metabolites were added to the urine. So you had two types of urine, spiked with metabolites or natural. These samples were sent out to 5 NMR laboratories and 10 mass spectrometry laboratories. We told the people, do what you do normally. Use the instrument as you normally do. Pre-treat the samples. Pre-treat the data that you acquire as you normally do. And so they sent us back data sets, tables, the lowest 88 variables and the highest almost 10,000 variables. So really everybody did it the way they normally did. I got the data and pre-treated it, Pareto scale for NMR, and then log 10 and Pareto for the mass. And here, this is just showing the number of variables in the different data sets. Sorry. Désolé. Right. The red ones, they're NMR instruments. So there's one with 88 and one with almost 10,000 variables. Wow. These are the mass spectrometers, and you had different technologies. There were 3 different technologies, and they have different numbers of variables as well. So I did ComDim on the results, on the data, and here I've plotted the scores of the individuals. So there were 15 people, 15 urine samples, either spiked or non-spiked. And common component number 1 perfectly separates out the 2 groups, without or with metabolites. And as you can see here, this is the importance of the table. This shows which laboratories, which technologies had an important effect to produce this dispersion. And you can see they all have an important effect. 3 NMR machines seem to be more important than most of the other instruments. A few mass spectrometers, 3 different technologies seem to have more importance than the others. But they all are fairly important. What's funny is the NMR machine with 10,000 variables isn't as good as the other ones, isn't as good as the one with 88 variables. But still, they all can detect this phenomenon. What's interesting to look at is common component 2. If you look carefully, you've got 1, 2, 1, 2 going up, another one, and a series. You've got exactly the same thing here. 1, 2, 1, 2 going up, another one, and then a series there. This is just showing, this is non-spiked and spiked. Just showing you can detect on all the laboratories, the NMR a little bit, not as well as the mass, can detect these differences between individuals. These are just variability from one person to another. Sort of thing you would expect to find. Common component number 3, this was a problem. Because when you're doing biological experimentation on human beings, even if it's just analyzing their urine, you have to sign some ethical documents saying that you're only going to use the data for the thing you plan to use it for, and you won't use the data for anything else. It's a serious constraint, and you can have serious problems if you don't obey these rules. And the problem is that there was one person who was very strange. And the guy organizing all this, he had a sort of moral problem, because if this person is sick, he shouldn't say anything. But if the person is sick, maybe he should say something. This is an ethical dilemma. There's no good solution. So he cheated a little bit. He looked at the list, and there was one woman in the group, and as well she was pregnant. So that's this woman here. So no problem anymore. Then you've got common component number 4. And this isn't a woman, and he's not pregnant. And so he thought about it for a long time, and he decided, right, it's a guy. We told everybody, do not go out and have a party the night before. Don't get drunk. Don't drink too much coffee. So they just supposed he was a guy. And so he didn't do what he was supposed to do. We don't know why he's different, but we hope it's just because he didn't do what he should have done. Okay, so you can see, coming back to more serious things, for this, all the laboratories can detect it to the same extent, except maybe this NMR here. These ones can all detect this. Here we can see that the mass spectrometry works better, but there's one NMR which is very good, and this sort of information can help the other laboratories to improve their methodology, their technology, to be up to this level here. But globally, you can trust metabolomics. I've got a little bit more time there. Okay. This is a study I did. People wanted to develop bio-sourced packaging material. And so, starch is bio-sourced, and it's something which is pliable. You can make shapes out of it very easily, but it's soluble in water. So for packaging, that's not too good. Lignin, on the other hand, is not soluble in water, but it breaks easily. It's hard to fold it. So what they were hoping was to be able to make mixtures of lignin and starch, which could then have better water-resistant properties and maybe be able to be used for packaging. So they produced 20 samples in duplicate, and They had 5 different lignin concentrations, 0 to 30. They extruded them, so they had cylinders, and then half of them they flattened, so they had cylinders and films. And then they put these cylinders and films at the 5 different concentrations in closed vessels at particular moisture levels, 33 and 75. And so we had, in the end, a whole set of samples, and I used a technique, I found many Brazilian colleagues here using this technique, time domain and MR, which characterizes the mobility of protons in the samples, different magnetic properties, nuclear magnetic properties of the protons in the samples. And so here I've got 8 different signals. This corresponds to solid and liquid. This set of 4 signals here, they can characterize the transfer of magnetization from the liquid phase protons to the solid phase protons. And here we've got an inversion recovery, which is always interesting to acquire. And this is one where I was hoping to get a T1 row characteristic. This inversion recovery gives you T1. These ones here give you T2s. So these are different characteristics of the relaxation properties of the protons. I didn't know which signal would be interesting, so I just acquired a whole lot of them and then analyzed the results. So I analyzed them using ComDim, 1 signal in each block of data. So I had 8 blocks of data, and I extracted 6 common components. And so here I've plotted the scores. So here we've got a packet at the top here and a packet at the bottom. This corresponds to the 2 moisture levels. So common component 1 is moisture. Here we've got oscillations like that. That's because we've got two cylinders, 2 films, 2 cylinders, 2 films. And here, common component 3, these are the different lignin concentrations, going from 0 up to 30%. So ComDim could extract these characteristics from the samples directly. Here we've got the saliences, the importance of the different data sets. So here, the one to look at first of all is common component 3. Only 1 data block is involved. The variability corresponding to the lignin concentration is explained by block number 7. And block number 7 is this one here. The inversion recovery characterizes the lignin in the samples. Here's these 2 blocks here, block 3 and 5, which correspond to this one and this one. These are parts of the signal that characterize the transfer of magnetization from the solid, the liquid to the solid. And so my theory is that since that corresponds to a cylinder and film, the creation of the film from the cylinders has changed the crystallinity of the lignin and the starch, and that has modified the transfer of magnetization between the liquid phase and the solid phase. And these ones here, all these different blocks here, contain information about the protons in the liquid phase. And here I've stuck the loadings of the 8 different blocks together. So here you've got the loadings of all 8 blocks, and you can see that's the loading for block 7. Here we've got the loadings for block 3 and 5, and here the loadings for nearly all the blocks, moisture, 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 etc. So ComDim can be used to see which blocks of variables are interesting. You can see if there are significant factors. You can use it later to create regression models to predict the lignin concentration, for example. And we've developed a method that allows you to do discriminant analysis as well, and even gone so far as to do a thing which is not very common in chemometrics, path modeling. You try and find the connections between all the different data sets. And we are thinking of using it to apply it to wavelet decomposition, apply it to 3D fluorescence, or even for color imaging. So we have a table for each color of your image. And I'm getting to the end there. When I do all this sort of stuff, I'm really enjoying myself. I find it's exciting because you can find so many things you didn't expect, and there are so many new ways of doing it. And what everybody should realize is this is only the beginning. We've got big data coming up. There is more and more data sets that are enormous and are going to need this sort of method. You've got artificial intelligence, which is going to make it all much more complicated. You've got the Internet of Things that people don't talk about anymore, but you can have things like your printer. I've got this. My printer automatically sends off a message to Hewlett-Packard to tell them to send me a new cartridge of ink. I don't even have to ask for it. It's done automatically because you've got the internet in the printer. You can have the same thing in your fridge. You can have the same thing in your washing machine. Once you've used up, before you've used up all your detergent, your washing machine tells Ariel or whatever to send you some more detergent. And this sort of thing can be in industry, pharmacy, agriculture, and then that can lead to things happening in chemometrics. Then there's lots of software being put into instruments, and this one I like in particular. This is a screen copy of my telephone. On my telephone, I've got MATLAB. This is a software, it's an incredible software. All my data analysis I do with MATLAB. Here, this is a little program written by some students in Campinas. They use your telephone to take color images of objects, and then they analyze this data using principal components analysis and other multivariate data methods. And then a last thing I want to talk about. If you are interested in using chemometrics, you want to understand how it works, but you don't want to program yet, there is the possibility of using a system called ChemFlow. This has been developed mainly by some of my French colleagues. We've been working on this for about more than 5 years, maybe getting on to 10 years. It's an internet chemometrics software. You don't have to download anything. You just have to have an internet connection and sign up through an interface to use this software. You can introduce your data. You can use different statistical methods, exploration, that's principal components analysis, and you can use ICA. This is one of the rare softwares where you can do ICA without programming. And you have very nice graphics. You've got explanations. You've got data sets that you can use to test to make sure you're doing things correctly. And you've got lots of information to explain how it works. And even better, these same people have created a MOOC, a massive online open course in chemometrics called ChemOOCs. It's the 6th year, I think, this has been running. We set this up because in France, there were only about 5 universities teaching chemometrics. Each one had a maximum of 20 students. That's only 100 people acquiring competence in chemometrics. It's not enough for all the different industries. They needed more. So we set this up, and now we regularly have 1,500 students signing up. Most of these students, at the end, do the exam and get a certificate saying that they're competent in chemometrics. And it's in 2 parts. The first part is non-supervised, like PCA and ICA, and the second part is regression methods, discriminant analysis methods, etc. It is free. It is really well done. You've got videos, you've got PDFs, you've got quizzes, you've got tests, etc. And they're working on a version in English, and in Spanish, and in Italian. Not Portuguese yet, but English. All the Portuguese people I know say they don't speak English. They say that to me in English. So I think as soon as we've got it in English, or if you want to brush up your French, I suggest you follow this. You sign up for this. It is really well done. And it's free, easy to have access to it. It requires about, this first module takes 7 weeks. And each week you have about, in all, there are 50 hours of videos and PDFs, etc. Over a period of 7 weeks, that's 7 hours a week. And in the end, you know a lot about chemometrics. Okay, I think that's all. Thank you. Obrigado. Perguntas or something like that.
Nesse momento, convidaremos o mestrando do Programa de Pós-Graduação em Ciências Farmacêuticas, Lucas Carg, para apresentar o nosso palestrante... Perdão. Na verdade, vamos convidar o Lucas para entregar um brinde ao nosso palestrante pela contribuição inestimável, Dr. Douglas Hutley.
For the photo. Muito obrigado. As they do in this one, I guess we have to do it this one. Obrigado. Muito obrigado. Muito obrigado a todos. I've had contacts with several different people. The main thing I'm doing at the moment is working with my colleagues Paolo and Vanessa, doing fluorescence, 3D fluorescence spectroscopy on wine. So we've done it on white wine, we've done it on red wine. What's interesting in wine is it contains different anthocyanins, different polyphenols, which give peaks at different positions of excitation and emission in the spectra, the fluorescent spectra. And what we want to do is to see if, using these 3D spectra, we can then differentiate the different varieties of grape, or the different regions where the wine is made, or the different years, etc. A trick that I've introduced, most people just do the fluorescence and then do multivariate data analysis on the fluorescent spectra. What I've been doing is I change the pH of the wine, because if you've ever drunk red wine and spilt a little bit on your white shirt, like me, you stupidly went and tried to wash it off with soap. Soap is basic, and when you put basic soap onto a red dot, a red spot, it goes blue. Why? Because you're deprotonating the polyphenols, the anthocyanins. That changes their structure and that changes their color properties. And so by changing the pH, we're changing the spectra, the fluorescent spectra, and getting interesting separations of the samples.
Outra pergunta?
Sim. I'm Rita. Congratulations for your talk. Cosimp is free, like the two last software that is.
Yes and no. Okay. I've got a toolbox, a Dropbox, and I've got all my programs in there. And anybody who wants it, I just send them the link, and they get all the functions. I update it regularly. I just updated it yesterday, changed a few things, but I just send it to anybody. But it's written in MATLAB, which means you have to have MATLAB. So my functions are free, but MATLAB isn't. But then on the other hand, it's a very good language, MATLAB. So if you can get it, and I also, there is another language, it's not Scilab, what's the, Paolo, do you remember the, he's not listening to me. He never listens to me. Octave, there's another language, Octave, which is free. And Octave is almost identical to MATLAB. You have to make very, very few changes, and anything you wrote in MATLAB, you can run in Octave. And in fact, for for ChemFlow, I write my functions in MATLAB, send them to my friends in Montpellier, who then put it into the program, just saying to the program, it's Octave. It's MATLAB, but he says it's Octave, and the program can then use it as if it was Octave. So there's very little difference between MATLAB, you have to buy, and Octave, which is free. And any of the functions you want, you can have them.
Can I ask some question? If I understand your talk, really, I have a doubt. Can I use chemometrics to unfold and refold peaks or macro data just from equipment like NMS or...
Which sort of data? MS? NMR?
Yes, if I generate my data,
Yep.
not using an instrument, but if I generate using information, DNA information, can I apply chemometrics to study disturbing and unfold and refold
but it depends on
signals?
It depends on whether you need to fold and unfold. Maybe with your data there's no need to. If you have a cube where each sample is a square or a rectangle, there you have to unfold. But if for each sample you've just got a line, there's no need to fold or unfold. If you've got a matrix already, you don't unfold. If you've got a cube or a hypercube, 4 or 5 dimensions, there you have to fold and unfold. And you can apply these techniques to any sort of data. I've used ICA on data about wine, sensory analysis on wine, and wet chemistry analysis on wine. And so I had the 2 types of data, and I applied ComDim to the 2 tables, sensory and chemical, to see the links between the different variables and the different blocks. So you can, any sort of data.
Thank you.
If it's good data.
Alguma outra pergunta?
Agradecemos a presença e contribuição inestimável do Dr. Douglas, trazendo brilho ao nosso evento. Muito obrigada.
Obrigado.
Aproveitamos para informar a todos os participantes que as atividades da 11ª Jornada de Farmácia Jofarma amanhã será realizada no auditório da CEAVEL. Na sexta-feira, voltaremos a utilizar esse auditório do CU. Na quinta-feira à noite, teremos a comemoração dos 25 anos do curso de farmácia, também neste auditório. A programação online do Congresso de Ciências Farmacêuticas do Mercosul e do Simpósio em Ciência e Tecnologia de Alimentos do Mercosul estarão disponíveis pela plataforma da EVEN 3 por um período de 60 dias para todos os participantes inscritos. Obrigada a todos que prestigiaram esta abertura. Tenham todos uma boa noite e um excelente evento. Todos estão convidados para o coquetel que será servido ali em frente ao auditório.